A couple of lectures ago, we stated that all viruses go through 6 stages during their life cycle: attachment, penetration, uncoating, biosynthesis, maturation, and release. Then we went through how influenza does each of these steps in part 3 of this video series. This video, part 4, describes how other viruses go through their viral life cycle. By the end of this video, you should be able to briefly describe what happens during the life cycle of minus-strand, plus-strand, and DNA viruses. You should also be able to describe the life cycle of HIV and explain what steps are targets for antiretroviral drugs. Finally, you should be able to describe how virus replication is different from binary fission. Let's start with something you're familiar with: the antisense or minus-strand type viruses. As I go through these life cycles, it may be useful to write down the differences between the viruses. You will also want to write down the examples of viruses that I've written in red. These are the ones that could show up on your exam. As you can see here, respiratory syncytial virus, influenza virus, and the viruses that cause measles and mumps are all minus-strand type viruses. They all have single-stranded RNA genomes, and as you'll recall from our last video, the fact that they're minus or antisense means that their genomes are like the complementary strand to a messenger RNA, and therefore they must be transcribed before biosynthesis can begin. So these viruses are not infectious on their own. They need the help of an enzyme called RNA-dependent RNA polymerase, and antisense or minus-strand viruses must bring a copy of that protein in their capsid in order to be infectious. Let's look at this generalized life cycle diagram to jog our memories about how the life cycle occurs. The first step of an infection with these viruses is attachment. Here, a spike protein or surface protein on the virus attaches to a receptor that could be a protein or a carbohydrate on the surface of a host cell. The binding of the spike protein to the receptor stimulates either endocytosis, fusion, or macropinocytosis. This will bring the virus into the cell in some sort of vesicle or membranous sac. Once inside the cell, so the virus has to uncoat. This will be step number 2. During uncoating, either due to the activity of enzymes from the host or the activity of acids which denature proteins in the capsid basically falls apart, releasing viral nucleic acids, in this case RNA, into the cytoplasm of the cell. Once the viral RNA is released inside the cell, infection can begin. The RNA-dependent RNA polymerase can then bind the RNA. Sometimes it's already bound to the RNA, but it'll start reading that RNA. It'll read the A's, the A's, U's, C's, and G's and make a complementary strand. In the case of a minus-strand virus, the complementary strand is a plus or a sense strand. Remember, the plus or sense strand acts like a messenger RNA. So that means that once this new viral genome is made, some of that can go out to the cytoplasm where host ribosomes can bind that plus strand RNA and start synthesizing viral proteins. The other thing that this plus strand RNA does is it acts like a template for making more minus strands. So there's 2 functions for the plus strand RNA in this life cycle. One, it serves as a template for making viral proteins. 2, it serves as a template for making viral nucleic acids. This is the next step, step 3 of your infectious cycle. This is biosynthesis. Step 4 is where we have translation of the viral RNAs to make viral proteins. So this is also part of biosynthesis. Okay, and then once we've got an accumulation of viral nucleic acids and proteins inside of the cell, they will spontaneously, somewhat spontaneously, begin to assemble to make new viruses. This is the maturation step. After new virus particles have been made, either at some of the in the nucleus, could be in the cytoplasm, could be at the plasma membrane. There's a number of places where this happens. But once the virus has assembled, it will bud away from the cell. That's release, the final step of the life cycle. Now remember, viruses that have an envelope are the ones that bud away from a cell. Viruses that do not have an envelope usually rupture the cell. They break it open, and this will release virus into the environment to continue the life cycle. Some viruses like poliovirus, rhinovirus, which is a major cause of the common cold, hepatitis A, and SARS-CoV-2, the cause of the 2019 global COVID pandemic, are sense or plus strand viruses. So remember, you wanted to write down the viruses that are in red as examples. For your plus or your sense strand viruses, the genome acts like a messenger RNA. These viruses are called infectious because as soon as they uncoat, host-derived ribosomes will bind their genome and start synthesizing viral proteins. I think it's only fitting, in view of what's going on right now, that we talk about the SARS virus as the model for this kind of life cycle. Coronaviruses are the largest type of plus-strand virus, with genomes ranging from 26,000 to 32,000 base pairs, and they measure approximately 125 nanometers in size. There are 7 types of coronavirus that are known to infect humans. Usually these viruses cause mild illnesses similar to that of the common cold, but 3 types, the SARS-CoV-1, that caused a pandemic in 2002, SARS-CoV-2, which is causing the pandemic from 2019, and MERS have all been identified as major causes of pneumonia in the last 20 years or so. SARS stands for Severe Acute Respiratory Syndrome, and MERS stands for Middle Eastern Respiratory Syndrome. All 3 of these viruses are enveloped, single-stranded RNA viruses. Their genomes encode up to 29 different proteins. 4 of those proteins are what we call structural proteins. So these are the ones that make up the virus structure. You probably want to know the spike protein, represented by the red triangles here. The spike proteins are proteins which radiate from the surface of the virus like a crown or a halo, hence the name coronavirus. Scientists think that a mutation in this protein gave SARS-CoV-2 the ability to jump from bats to humans as hosts. The spike protein is also a major target for vaccine development. Labs around the world are working on molecules that can prevent spike protein binding its receptor or stimulate our immune systems to produce antibodies that will bind the spike protein and disrupt its ability to bind our cells. By the way, despite what certain political figures say, as of the recording of this video in the spring of 2020, we are probably 6 months or more away from having a vaccine that is widely available. Other structural proteins are the membrane protein, which helps to form the outer surface of the virus. You have an envelope protein, which helps form the lipid layer, called the envelope, around the virus. And then you have the nucleocapsid proteins, called N proteins. They bind the viral RNA. Non-structural proteins are proteins that are involved in the virus infection, but they don't form the capsid or the envelope of the virus. Genes for these proteins make up the rest of the viral genome. I won't go through all these proteins, but I do want to mention a handful to give you an idea of what kinds of things non-structural proteins do. Proteins like NSP1, NSP3, NSP10, NSP15, and NSP16 defend the virus against detection by infected host cells. They weaken the cell's ability to ward off infection with antiviral proteins. They disguise the viral RNA so that cellular antiviral proteins can't recognize them. And in some cases, they actually chop up little bits of viral RNA that don't get incorporated into viruses so that it's harder for an infected cell to display those strange molecules on their surface and be recognized as infected by the immune system. NSP7, NSP8b, NSP12, and NSP14 form a replication transcription complex. So NSP12 is the RNA-dependent RNA polymerase. And then these other proteins are like helpers. You can think of them as being like a viral copy machine. This complex reads the viral genome and makes a complementary copy. NSP14 is a proofreading enzyme. So if the NSP12 protein, that polymerase, makes a mistake, 14 recognizes that mistake, cuts it out, and then allows NSP12 to put the proper nucleotide in its place. Do I expect you to remember the names of all these proteins? Oh no! I would just like for you to be aware somewhere in your minds that although viruses are small bits of nucleic acid and protein, they can really be quite sophisticated. I do expect you to remember the general plus-strand viral life cycle, which we'll talk about on the next slide. Now let's look at the life cycle of SARS-CoV-2, as this is pretty standard for a plus-strand virus. Infection begins when a spike protein on the surface of SARS-CoV-2 binds a receptor on epithelial cells in the body. This receptor is called ACE2, or the angiotensin converting enzyme 2 protein. We find this receptor on the surface of cells in the alveoli, in the trachea, in the bronchi, and in the bronchial serous glands. But that's not all. It's also on the alveolar macrophages. We find it, believe it or not, in cells of the gastrointestinal tract, cerebral neurons. We find it on endothelial cells in the arteries and veins. It's even on cells in the renal tubules. Does the virus infect all of these cells? Potentially, yes. We know that coronaviruses can be shed not only in respiratory droplets, but also through urine, stool, and sweat. Okay, so back to the diagram. SARS-CoV-2 binds its receptor ACE2, and this triggers endocytosis into an endosome. Remember, an endosome is kind of an acidic bag inside the cell membrane, or inside the cell cytoplasm, rather. The acidic environment of the endosome causes structural changes to the spike protein and permits the fusion of the viral envelope to the endosome membrane. This releases the virus into the cytoplasm. Now, at the same time as this is going on, the capsid that surrounds the nucleic acids of the virus begins to fall apart, so that it's truly the nucleic acids that get released into the cytoplasm. Now, since this is a plus strand or a sense virus, the genome acts like a messenger RNA. So that means that host ribosomes, represented by the bubbles that you see here, host ribosomes bind that RNA and start translating two polyproteins immediately. The best analogy that I can think of for polyprotein is those toys that come in fast food kids meals, where you have all the pieces in this one thick piece of cardboard or plastic. In this form, the parts of a toy won't make an airplane, a house, or a car. They're really hard to play with. But if you punch the pieces out or separate them and then assemble them into a functional toy, well, that's a lot more fun, right? And it has more function. Polyproteins are like a collection of proteins that are all tied together, and they're non-functional in this form. A protease, which you can think of as being like molecular scissors, has to come in and separate the proteins for them to serve their function. One of the proteins that is liberated by the protease for SARS is the virus's RNA-dependent RNA polymerase. And then there are some associated proteins like that NSP14 that get released as well. Together, the RNA-dependent RNA polymerase and its support proteins read the plus strand viral genome and start making minus strands. Now, these minus strands have a special function. They are going to be templates for making more plus strands. From the plus strands, more viral proteins can be made because remember, the plus strand acts like a messenger RNA. As viral proteins begin to accumulate inside the cytoplasm of the cell, so here are the different transcripts that can be made, and those get made into proteins. So as those proteins are made, they actually get inserted into the lumen of the ER. And they're going to move from the ER through the Golgi, Golgi intermediate compartment. So that's what's going on here. You see the S, for example, that's the spike protein. M, that's the membrane protein. E is the envelope protein. They start out here in the ER and they're moving through the Golgi. They'll eventually bud off into vesicles, and this will serve as the site for virus assembly or maturation. As these vesicles move through the cytoplasm, they're going to bring mature viruses to the plasma membrane where the virus will get exocytosed. It'll bud away. So what you need to remember is this basic diagram. Again, the virus is going to attach to a host cell through some receptor, and it'll be brought into the cytoplasm, again, by endocytosis, fusion, or macropinocytosis. The virus must uncoat. This will release the viral genome into the cytoplasm of the cell. Now, one difference between your sense strand virus and your minus strand virus is, remember, your sense strand virus acts like a messenger RNA. So cellular ribosomes can bind to that viral RNA and start synthesizing proteins. Remember, all this virus needs is a gene, because the host can immediately start synthesizing the RNA-dependent RNA polymerase necessary for replication of the virus. This is different from the minus strand. Remember, the minus strand had to actually bring the protein itself in the capsid. Why? Because hosts don't make that enzyme. So there's no enzyme to be had for the minus strand virus unless it brings the protein with it. So this is a big difference. All right. Once the host ribosomes start synthesizing that RNA-dependent RNA polymerase for the sense strand virus, that polymerase will start synthesizing minus strands, and this is through transcription. Those minus strands serve as templates for making more plus strands, and then the plus strands go on to be templates for making viral proteins. So what we're going to do is start accumulating viral proteins and accumulating viral nucleic acids together during a step of biosynthesis. So that's what's going on here. Once there's enough of this material inside the cell, the molecules, the RNA and the proteins will start assembling together to make new viruses. This is maturation, and that new virus will be exocytosed. That's called budding. Just like we said with the sense strand viruses, budding happens with your enveloped viruses. Viruses that don't have an envelope usually rupture the cell. Retroviruses are a unique group of enveloped RNA viruses because they spend part of their life cycle as DNA viruses. Currently, there are 6 known retroviruses that infect humans, but only 4 of those are associated with disease. Perhaps you've heard of them: HTLV1 and 2 and HIV1 and 2. The 2 remaining retroviruses are called HTLV3 and 4, and they were discovered in 2005 but are not known to cause disease as of this video recording. All right. HTLV1 and 2 were the first retroviruses discovered, but we now know that similar viruses infect a number of primates. HTLV stands for Human T Lymphotrophic Virus, or Human T-cell leukemia lymphoma virus. These pathogens infect human T cells. HTLV1 causes a rapidly developing and often fatal leukemia in 4 to 5% of infected adults. It's also known to cause debilitating neurological problems and infectious dermatitis. HTLV2, on the other hand, causes milder neurological problems and is associated with chronic lung infections. Both of these viruses are transmitted through sexual contact or blood-blood contact. So think about, for example, people receiving blood transfusions, or maybe they're intravenous drug users, or maybe they are healthcare professionals who get a needle stick. Okay. HTLV1 and 2 can also be transmitted by breastfeeding. HIV stands for Human Immunodeficiency Virus. Like HTLV, HIV infects CD4 cells. So I don't think I mentioned that a minute ago, but both of these viruses infect a special kind of immune cell called a CD4 positive T cell. These T cells are also called T helper cells. If you think of the immune system as being like an army, T helper cells are like the generals. They direct the response of the immune system to pathogenic effects. Remove the T helper cells and a person's ability to fight off disease and cancer is severely impaired. When left untreated, HIV causes acquired immunodeficiency syndrome, also called AIDS. AIDS has a number of signs and symptoms associated with it, but low T cell counts, swollen lymph nodes, thrush, diarrhea, reoccurring opportunistic infections, and a type of cancer called Kaposi sarcoma are common. HIV is transmitted by sex, blood-blood contact. So again, these are the needle sticks and the blood transfusions, okay, and also breast milk. And just to clarify, when we talk about sex here, primarily we're talking about anal sex or vaginal sex. The risk of infection is much lower for oral sex, although there is some risk. Let's take a look at the life cycle for HIV. Now would be a good time to pause this video and watch the video HIV Virus Life Cycle animation from the lecture video folder. So again, there are 6 strategies or 6 locations where antiretroviral drugs can interrupt the HIV life cycle. The video Treating HIV, Antiretroviral Drugs, Infectious Diseases, NCLEX RN, Khan Academy in your lecture video folder goes through each of these. Now would be a good time to view that. But in case you'd rather wait until chapter 20 to look at all 6 strategies for this exam, you really only need 3. The three strategies that you need to know for this exam are, number one, the nucleoside-nucleotide reverse transcriptase inhibitors, or NRTIs. These are nucleoside analogs that inhibit the conversion of RNA genomes into DNA. What they do is they compete with guanine nucleotides for incorporation into viral DNAs. Once these analogs are incorporated into DNA, they prevent transcription and reverse transcription of the viral molecule. Protease inhibitors are another class of drug that prevent the final step of maturation. So if viruses are produced, they are non-infectious. The final drug that you need to know is the integrase strand transfer inhibitors, or ISTIs. These block infection by inhibiting integrase activity. Remember, integrase is the enzyme that incorporates the viral genome into the DNA of T helper cells. Now let's talk about the last group of viruses for this exam, the DNA viruses. DNA viruses have a double-stranded DNA genome, all except for parvovirus, which is single-stranded. The DNA genome of these viruses is replicated by host DNA-dependent DNA polymerases. Some of your larger viruses, though, will bring their own viral DNA polymerases to replicate their genome. Your DNA viruses are grouped into three groups based upon how big their genome is. You have the small viruses, which have genomes that are less than 10,000 base pairs, and your polyoma viruses and human papillomaviruses fit into this category. You have medium-sized viruses that have a genome that's about 35,000 base pairs. Adenovirus would be an example of this. And then you have some viruses that have really large genomes, like 150 to 250,000 base pairs. Herpes viruses are pretty much the only viruses in this group. Another thing that's common among your DNA viruses is that their genome, their genes, are divided into early and late. Early genes are genes that are related to genome replication, and late genes are usually genes that are related to the virus structure, like capsid proteins. We will use human papillomavirus as our example of a DNA virus. There are over 100 different types of human papillomavirus, and they cause a variety of different diseases, from warts that you might find on your skin to cervical cancer. The virus enters the human body via damage to your epithelial layers. This allows capsid proteins on the virus to bind to heparan sulfate proteoglycans on the host's cell surface. Heparan sulfate would be the receptor for this virus. Once the capsid proteins bind to the receptor on the host, the virus is endocytosed by receptor-mediated endocytosis. Once the virus comes in via an endosome, this acidic environment causes the capsid to fall apart, delivering the genome to the cytoplasm. The genome for the virus has to be transported to the nucleus, where it gets maintained as an episome, or as a circular bit of DNA that's outside of the host chromosome, kind of like a plasmid. I've provided a video in your lecture videos folder called Natural History of HPV Infection that describes the HPV life cycle and explains how the virus causes cervical cancer. Now would be a good time to pause this video and watch that one. So, in addition to HPV, varicella zoster, which is the virus that causes chickenpox and shingles, your herpes viruses, adenovirus, and hepatitis B are all DNA viruses. The DNA virus life cycle, really briefly, occurs in the following steps. Interaction between a viral, either spike protein or capsid protein, and a receptor on a host cell initiates the infection through the first step called attachment. The virus then enters the cell via endocytosis or some other mechanism, and the virus must uncoat. This can happen via the activity of enzymes from the host cell or acidification of the capsid, and that causes the capsid to fall apart, releasing the viral DNA into the cytoplasm of the cell. Now, what makes this virus different from your other viruses is that this DNA has to be replicated inside of the nucleus. So that means that it must be transported to the nucleus. Once that DNA from the virus is inside the nucleus, the host cell's DNA dependent RNA polymerase can read the genes on that DNA and start transcribing messenger RNAs. These messenger RNAs can then travel to the cytoplasm of the cell, where they can be bound by host ribosomes, and capsid proteins and other structural proteins from the virus can be synthesized. Some of those proteins may be transported back into the nucleus where they'll bind to the viral DNA, and then that DNA protein complex gets transported back to the cytoplasm for assembly of the virus. For some of your DNA viruses, assembly may actually happen inside the nucleus. Viral release happens when the virus either buds away from the cell or when it ruptures the cell. Usually, cell rupture happens when the virus triggers the host cell to produce some proteins or enzymes that destabilize the plasma membrane, and that causes lysis. Or sometimes it's just the proteins that the virus makes during its replication cycle that are actually toxic to the cell, and they accumulate as occlusion bodies, or they actually trigger the cell to self-destruct. Whoa. That was a long video, but hopefully the viral life cycles are a little clearer now. If you need to, go back and watch part 3 and part 4 of this series again, because there will be questions related to all of the virus life cycles on your next exam. During this video, I described what happens during the life cycle of minus strand, plus strand, DNA, and retroviruses. I hope what you took away from the life cycles is that all of the viruses go through the same basic steps, although they accomplish the steps differently. I've tried to highlight how these viral life cycles vary in the table at the bottom of this slide. I hope it's helpful. Hopefully you watched the videos about HIV and antiretroviral drugs from the lecture videos folder. Isn't HIV's life cycle unique? You'll need to remember the steps of HIV's life cycle and also 3 strategies for treating HIV infections. Remember, there are reverse transcriptase inhibitors. These are molecules that look like RNA nucleotides, but they stop transcription and reverse transcription. You have and remember, reverse transcription is the conversion of HIV's RNA genome to DNA. You have integrase inhibitors, which are molecules that inhibit or interfere with the insertion of HIV DNA into host chromosomes. And finally, you have the protease inhibitors that prevent the cleavage of HIV polyproteins and inhibit virus maturation. Because parts 3 and 4 of this series were such long videos, I did not include review questions here, but I will post a set to your course space as a practice test or a separate video. Until then, keep up the good work. If you have any questions, don't hesitate to reach out to me. I'll see you next time.